Today, we'll be doing a mind map on molecular biology. Multiple molecules are essential for living things to function, and I'll be discussing these molecules today. Firstly, we have carbohydrates, which are biomolecules containing carbon, hydrogen, and oxygen. The smallest carbohydrates are monosaccharides, also referred to as sugars. A monosaccharide you may have heard of before is glucose. Glucose exists either with an alpha-D-glucose structure or beta-D-glucose structure. The configuration only differs in the positioning of the hydroxyl group at the carbon-1 atom. Other monosaccharides you will have to be familiar with are fructose and galactose, though there are more monosaccharides. All three monosaccharides have the molecular formula C6H12O6, though in differing arrangements. Monosaccharides always have the carbon, hydrogen, and oxygen in the ratio of 1:2:1. These monosaccharides commonly link together to form the following disaccharides. Glucose plus glucose forms maltose, glucose plus fructose forms sucrose, and glucose plus galactose forms lactose. This occurs through a condensation reaction with water as a byproduct. Carbohydrates are built up by condensation and broken down by hydrolysis. When two monosaccharides join together, hydroxyl groups on each molecule react to form a bond, losing a molecule of water in the process. This bond is called a glycosidic bond. This process is a condensation reaction, while the opposite is called hydrolysis. Both reactions are aided by enzymes. When multiple monosaccharides join to form glycosidic bonds, they form polysaccharides. The three polysaccharides that you will need to know are starch, cellulose, and glycogen. All three are polymers of glucose. Starch is the stored form of sugars in plants, cellulose is found in cell walls, and glycogen is a stored form of sugar in mammals. A common essay question is the comparison of polysaccharides amylose, glycogen, and cellulose in terms of their similarities and differences. Amylose is one form of starch, with the other being amylopectin. However, amylose is often used for comparisons. So firstly, amylose and glycogen are made up of alpha-D-glucose molecules, while cellulose is made up of beta-D-glucose molecules. The bonds between glucose molecules are only between the carbon-1 and carbon-4 molecules for amylose and cellulose. Glycogen, on the other hand, has both 1,4 linkages and 1,6 linkages. This makes glycogen a branched polymer, while amylose and glycogen are unbranched. Amylopectin is similar to glycogen in that it is also a branched polymer of alpha-D-glucose, having both 1,4 and 1,6 linkages. The function of amylose is the storage form of sugar in plants. Glycogen is the storage form in mammals, and cellulose is essential for cell structure. In terms of shape, amylose has a curved shape, which gives it a helical structure. Glycogen has a bent and compact shape, which gives it a branched out structure. And cellulose adopts a straight chain shape, which gives it a layered structure. In terms of transport, amylose is transported in the form of sucrose, glycogen is transported as glucose, and cellulose is transported as part of the cell wall. All three are also insoluble in water. In comparison of strengths, glycogen is the weakest, partly due to its branched out structure. This is useful though, as it allows glycogen to be quickly hydrolyzed to glucose in the body. Following this is amylose and cellulose, which is the strongest polysaccharide out of the three. This is due to the layered structure of cellulose, which allows for hydrogen bonding to occur between and within cellulose molecules. This hydrogen bonding makes cellulose much stronger than the other two, whose structure prevents the formation of such hydrogen bonds. Speaking of hydrogen bonding, we move on to a very important and interesting biological molecule, water. In liquid water, hydrogen bonding occurs between water molecules due to the polarity of water. The structure of water explains its polarity. In water, one atom of oxygen is bonded to two separate atoms of hydrogen via a single covalent bond. However, due to the stronger attraction of the oxygenous nucleus, there is an uneven distribution of electron density about the molecule. This causes part of the molecule to have greater electron density and less electron density, which creates a partially negative pole and partially positive pole respectively. These partial charges make water a polar molecule, which easily forms intermolecular hydrogen bonds. This bond occurs between the partially positive hydrogen and the partially negative oxygen of separate water molecules. The importance of this is that the polarity of water gives it properties that make it useful for biological systems. Water has a high melting and boiling point, which allows it to exist as a liquid in most habitats, which is essential for living organisms to survive. Water also has a high specific heat capacity, which means that its temperature does not fluctuate erratically. This helps maintain stable environments, with an example being human bodies, which is 60% water. Body temperature is therefore regulated and relatively constant, which is important for chemical reactions in the body to take place. Water also has a high latent heat of vaporization, which makes it an effective coolant. An example of this is us sweating to remove heat. Apart from its thermal properties, the polarity of water makes it a good solvent. This is very important for many processes in the body such as gaseous exchange and multiple metabolic processes in which water acts as a medium. An example of an important process in which water acts as a solvent is respiration. Other processes in which water is essential include hydrolysis, which we just discussed, and photosynthesis. The polarity of water also causes molecules to cohere to each other and adhere to polar surfaces. On top of increasing the strength of bonding, it is very important in the drawing of water-up xylem vessels in plants. This will be discussed in my Mindmap on plant structure. The next biological molecule we are going to discuss is lipids, commonly referred to as fats. There are three types of lipids: triglycerides, phospholipids, and steroids. When talking about lipids or fats, it usually refers to triglycerides, where phospholipids and steroids will be referred to as such. A triglyceride lipid molecule is made up of glycerol and three fatty acid chains that undergo condensation. The hydroxyl and carboxyl groups of the respective molecules react to form an ester bond, producing water as a byproduct. Phospholipids differ where it only has two fatty acid chains linked to glycerol, with a phosphate group bonding with the third hydroxyl group of glycerol instead. All steroids have a basic skeleton of 4 fused rings. Lipids can be identified by these defining structures. You may have heard of saturated and unsaturated fats before. This refers to the bonding present in the fatty acid chains that make up lipids. The difference between saturated and unsaturated fatty acids is the presence of double bonds between carbon atoms in the chain, where saturated fatty acids do not have carbon double bonds, while unsaturated fatty acids have at least one. Mono and polyunsaturated fatty acids refer to whether there is only one or more than one carbon-carbon double bonds respectively. Another comparison you will come across is cis-unsaturated and trans-unsaturated fatty acids. Cis-unsaturated fatty acids have hydrogen atoms bonded to carbon atoms on the same side of the double bond, while trans-unsaturated fatty acids have hydrogen bonds bonded to carbon atoms on opposite sides. In these two comparisons, the basic rule of thumb is that unsaturated and cis fatty acids are healthier. This is because trans fats cannot be metabolized and when left in the body will affect membrane fluidity. However, there is more detail in terms of the different health risks and effects associated with lipids. This is something you can look more into as it can be part of a nature of science inquiry or a data interpretation question. The main functions of lipids are for energy storage, insulation, and protection of organs in the form of adipose tissue. The final biological molecule is protein, whose smallest unit is amino acid. All amino acids have the following base structure. Around a central carbon atom, there will be a carboxyl group, an amine group, a hydrogen atom, and an R group. The R group represents the different variations an amino acid molecule can have, where there are 20 variations, corresponding to the 20 amino acids that exist. These R groups can have various properties, such as being polar or not, being ionic or not, or containing sulfur. Just like carbohydrates, amino acid molecules undergo a condensation reaction to bond with each other and form an amino acid chain. Chains of less than 40 amino acids are called peptides, and chains of more than 40 are called polypeptides or proteins. The amino acid chain is the primary structure of proteins, where amino acids are bonded to adjacent amino acids via peptide bonds. This process occurs at ribosomes in cells, as discussed in my Mindmap on cellular structure. Proteins then can go on to form secondary structures, with the major ones being alpha helixes and beta plated sheets. In these structures, amino acids form hydrogen bonds with other amino acids in the chain, where these amino acids may or may not be adjacent to each other. Proteins subsequently may form tertiary structures due to further interactions between amino acids that gives it a more complex structure. Some interactions include the formation of disulfide bridges, ionic interactions, hydrophobic interactions, and additional formation of hydrogen bonds. These interactions occur mainly between R groups of amino acids. However, it may also exist with other parts of the amino acid, as is the case of formation of hydrogen bonds. Hydrophobic interactions usually occur in the middle of the protein and is what creates folds. When proteins form tertiary structures, they would have formed a 3D structure. Quaternary structures form when multiple polypeptides interact with each other. This is the case for protein complexes, to give an example. There are way more things to talk about with regard to proteins and its applications. Therefore, I'll be doing a separate Mindmap on proteins that you can check out, because what we have discussed so far is mainly the basics of protein structure. Another important biological molecule is DNA and RNA. However, I have a completely separate Mindmap for them, which you can check out. This brings me to the end of my Mindmap on biological molecules. Hope you find it useful and have a good day.